All right, welcome back. We're going to continue with Chapter 5, and this time we are going to start looking at proteins. Okay. So Concept 5.4, proteins include a diversity of structures resulting in a wide range of functions. Okay. So proteins account for more than 50% of the dry weight of most cells. So most things in your cell are proteins. Some of these proteins result in speeding up of chemical reactions. We refer to those as enzymes, and I'm sure you've heard of those before. But proteins can do a lot of other things as well, including defense against microorganisms. It can be for storage, transport, cellular communication, movement, structural support, you name it. There's probably a protein that can do it. Okay. So, enzymatic proteins are essentially something that can accelerate a chemical reaction or act as a catalyst. It does this by essentially taking two things that, in this case, need to be broken apart. It binds it to the surface of this enzyme, and then what probably happens is the enzyme facilitates a disruption of this bond, either by pulling these two pieces apart or maybe introducing a bond that replaces one of these bonds that is then broken. There's lots of different ways to achieve this, but then each individual piece can fall out. And this is typically how an enzyme works. It will bind the various things that it needs to react, and it will put it into a physical arrangement that makes that chemical reaction likely to happen. Now, these enzymes can be used over and over and over again, so one enzyme can produce lots and lots of products. We have defensive proteins. So here is an example of antibodies. Antibodies are a protein that can recognize specific viruses or a specific bacteria. When you coat that virus or bacteria with antibodies, it can lead to either inactivation of those or labeling those microorganisms for destruction. Proteins can also act as a storage for amino acids. So when you start thinking about things such as eggs, where all of the material for that chick needs to be contained within that egg, you need sources of amino acids. In eggs, we have something called ova-albumin. That is essentially a protein whose sole purpose is to store all the amino acids. You break this down in order to liberate amino acids, which then go to build the proteins that the embryo actually needs. Same thing holds true for plants. Plants have seeds and they have storage proteins that act the same way. For mammals, we have this protein called casein in milk, which serves as an amino acid source for babies or offspring. Proteins can be involved in transport. So hemoglobin is something in our blood which transports oxygen throughout our body. Or you could have things that are proteins that exist in membranes, and their job is to transport an ion or some molecule across the membrane, right? So it can be involved in transporting other things. You can have proteins that act as hormones. So after we eat a meal and our blood sugar starts to rise, we need a signal for our body to absorb all of that sugar. That signal is a protein called insulin. It's secreted. That tells all of our cells to start absorbing that blood sugar and put it into storage within the cells. We have receptor proteins. So here we have the surface of one neuron, and there is this receptor protein on the membrane. And here is another neuron, and what it's doing is releasing compounds that tell the next neuron to fire. Something has to be responsible for recognizing them. That is these receptor proteins. They bind this molecule, whatever it happens to be, and then they result in some type of signal happening within the cell. We can have contractile and motor proteins. So here we have muscle tissue. Muscle tissue is made up of these large fibers called actin. Those tend to be somewhat structural in nature, right? They're just essentially long fibers. Here we have myosin, which acts as a motor. It has the ability to bind and move along an actin filament. So its ability to walk along these actin filaments in either direction pulls the two ends closer together, and that's muscle contraction. This is just one example of the motor aspect of some proteins. We can have structural proteins. Here we saw an example of this with actin. So actin itself is a structural protein, to where myosin is the motor protein. But we also have something in our skin called collagen, which is kind of these three long fibrous proteins wrapped around each other. This is what makes our skin strong. This is what allows us to have a dead layer of skin on the outside of our body that doesn't fall apart. This is also what makes up hair, makes up horns, makes up feathers, nails, all kinds of things there. You can also see structural proteins in spider silk. So that is a protein. So you see this in connective tissue as well. So here we have proteins acting in a structural manner. Now, proteins are all built from the same set of approximately 20 amino acids. We refer to these long structures as polypeptides. Okay, so they are long unbranched polymers built from individual amino acids. We usually reserve the name protein for something that has some type of biological function. So sometimes you can have a polypeptide that is a precursor to a protein. So something happens to activate that polypeptide, activates its activity, and turns it into a protein. But in reality, just keep in mind that proteins are in fact polypeptides that just simply have a function. Now, amino acids are the monomers in this, and they have both an amino group and a carboxyl group. They also have this side group called an R group, and that R group is what can give each amino acid a different property. So here, looking at a picture of it, we have a central carbon. We call that the alpha carbon. On one side, we have an amino group. On the other side, we have a carboxyl group. Again, an amino group can absorb a proton and become positive. Carboxyl groups can donate a proton and become negatively charged. On one side of the alpha carbon, we have a hydrogen, and then we have some type of R group. And there are plenty of different R groups that come in all different types of flavors and activities. Okay? We can break this up into 4 large groups. So we have hydrophobic side chains and polar side chains. Within those polar side chains, we may have polar but uncharged. We may have polar and negatively charged, so things that are acidic in nature. Or we could have side chains that are basic in nature, and they will be positively charged. So let's go through each one of these groups. So here we have all of the nonpolar. You can see it can start off as simple as just simply a hydrogen. We can then attach a methyl group. We can make that methyl groups longer. We can rearrange them. We might have sulfur present in some of these. We may be forming various rings. We can even have a long hydrocarbon chain that wraps around and reattaches to the chain. So this is proline. So the side chain actually reaches around and binds with the nitrogen. Each one of these has a different characteristic, and we will talk about those later on. For the polar side chains, the uncharged ones, we have serine, which is simply a hydroxyl, threonine, which has a hydroxyl but also has a methyl group. We have cysteine, which has a thiol group. Again, thiol is somewhat similar to a hydroxyl because sulfur is similar to oxygen. Here we have tyrosine, which looks a lot like phenylalanine. It just has a hydroxyl attached. Here we have asparagine and glutamine. They both have carbonyls and they also have amino groups. For the acidic, these all have side chains which have carboxylic acid or carboxyl groups. These can ionize and at physiologic pHs are typically negatively charged because of that. The basic ones include lysine, arginine, and histidine. They all have various amino type groups that are basic in nature, so they can absorb a proton and become positively charged. Some of these various amino acids we should talk about separately as well. So they tend to be special in nature. Glycine is one example of that. So glycine itself has a very small R group. And you will also notice here that this carbon is not chiral in nature because both of these are hydrogens. This makes glycine very flexible. Proline, on the other hand, has a side chain that wraps around and touches and binds covalently to the nitrogen. Ordinarily, you get free rotation between this nitrogen and this carbon, and between this carbon and the carboxylic acid carbon. That cannot happen, and it actually causes a kink to occur in the chain. Consequently, because this is so flexible, because this forms a kink, you oftentimes see glycines and prolines in a sharp turn that might occur in a protein. We have one amino acid called cysteine that has a thiol group on it. Something interesting that can happen is if you have two thiol groups that are opposite each other, they can actually react and covalently bond with each other. You'll see we lose the hydrogens in this reaction. And this combination we actually call a cysteine. Another word for this is a disulfide bond. This can be a strong stabilizing force in a protein.